Are we all set? Yeah. Well, welcome everybody. First of all, hello. I'm Tim Angley, as it says, and I work at within the GI research group here at the ABI. So quickly, what are all of your backgrounds? Obviously, you're all new doctoral students with the MedTech core. And where are you from? What are your backgrounds? Is it engineering primarily?
Mechanical engineering.
Mechanical? Bioengineering? Engineer? Physiotherapy, okay. Mechatronics?
Engineering and neuroscience.
Okay, good. So we've got a nice varied background. So what I'll do is I'll give you quite a general overview of GI activity. I'll explain everything underlying it and talk about some of the different applications that we've developed in terms of engineering techniques, mathematical modeling, experimental techniques, and also some clinical translation that we've achieved and are continuing to achieve. So our group is primarily focused in gastrointestinal electrical activity, and that's important because each of the underlying contractions in your GI tract is initiated and coordinated by an underlying electrical event. So it's very similar to cardiac electrophysiology. We compare it to a lot, where your heartbeat is initiated and coordinated by an underlying electrical wave. So here we can see recording from inside the stomach. So this is an endoscopic view. A camera is placed inside the stomach, and we can see these large ring contractions propagating down the stomach towards that distal end of the stomach. And each of these contractions is initiated and propagated by an individual electrical wave termed the slow wave. Now, obviously, these contractions are vital for the breakdown and movement of contents through your digestive tract. They underpin the process of digestion and the whole coordinated process of the GI tract termed gastrointestinal motility. And it's these individual electrical waves or electrical events that we are interested in studying as one of those key mechanisms underlying digestion. So to cover a bit of anatomy to start, first of all, in terms of gastric anatomy, because I'll focus primarily on the stomach for this talk. Now we do some work in the esophagus on the top end and the small intestine and large intestine more distal, but primarily I'll focus on our gastric work. So in the stomach you have your esophagus, which comes down from your mouth down and joins the stomach at the top end. And you've got your small bowel that leaves the stomach at the bottom end and is separated by a sphincter called the pylorus or the pyloric sphincter. So just a bit of terminology for the rest of the talk. And then your stomach is separated into three individual anatomical units, although they're not actually physically separated. So the top of the stomach is called the fundus. That's primarily serves as a storage reservoir for food, accommodates to hold food. The middle of the stomach is called the body or the corpus, and that's where we get a lot of our mechanical breakdown of the contents of the stomach. And the distal stomach is called the antrum, and that again is where we get a lot of mechanical breakdown, but also movement of contents through the pyloric sphincter or pushing back up into the stomach to continue to break down those contents. Now, from an electrophysiology point of view, one of the very important cell types is called the interstitial cells of Cohort. They're a specific cell type that's located between the layers of smooth muscle, primarily, but also distributed between the smooth muscle. So if we look at the gastric wall here, we have two layers of smooth muscle, one that runs longitudinally down the stomach and one that runs circumferentially around the stomach. These ICC cells are located primarily between those smooth muscle layers, but also distributed throughout the smooth muscle layers as well, which we can see a little bit better in this cellular or tissue level image here. So what we see here is a cross section of the gastric wall, and we can see the two smooth muscle layers stained green. So we don't know which direction either one is, but you can see they're running transverse to each other. So we've got one smooth muscle layer running this way and one smooth muscle layer coming into and out of the screen. And then we can see our ICC cells have been stained red here. So we see that they're primarily located in a dense layer between the smooth muscle layers, but also distributed throughout. And it's important because these ICCs have been identified as being the primary cell responsible for not only initiating slow wave activity, but also propagating it. And we can see an example intracellular recording down here, where we see that the ICC activation precedes the SMC or smooth muscle cell activation. And this is important because we've learned that ICC are responsible for not only the generation and propagation, but that the SMC then can only passively propagate or passively conduct that activity. Now, previous studies of slow wave propagation, particularly at the organ level, have utilized what we term as low resolution techniques, where a few electrodes were sparsely placed along the length of the organ, and individual electrode recordings were obtained from each of these electrode sites. So we've got a little example here, 4 electrodes and example electrode traces. But the problem with this, or with these data, are that it forced researchers and clinicians to make assumptions about the activity occurring between these electrode sites, because there wasn't enough spatial resolution to directly join the activity from one electrode to the next. So in reality, we may think that these individual activation points or slow wave events are linked in sequence as shown on the screen. But it may actually be that there's a time delay, and this event correlates with this secondary event and this tertiary event in the third channel and further down along the line. So these, I guess, low resolution techniques, while they form the foundation of our field and gave us very important information about slow wave characteristics like the frequency of events at individual sites, it precluded any sort of spatial analysis of slow wave propagation patterns. Now, another type of, I guess, historic or still current GI electrical recordings are body surface electrogastrography, or EGG as they call it. So this is similar to, in cardiac electrophysiology, your body surface ECG, or in neural recordings, the EEG. So in this case, we place a number of electrodes on the abdomen of the patient, directly over or roughly over the location of the stomach, and we record those body surface potentials. And there's an example shown down here. Now you'll notice, although these are just example recordings, here our example looks very nice with individual events, and here we see a very noisy signal. Very difficult to find any sort of routine activity at a frequency of around 3 cycles per minute, which is how fast our slow waves propagate. So one of the issues with these signals is that they're prone to noise, they're prone to picking up all of the electrical interference or artifact from the organs that we're not interested in when we're looking for specifically, say, stomach activity. We pick up interference from real high amplitude cardiac, or relative compared to our gastric signals, very high amplitude cardiac signals, small intestine signals that are all within that same region, colonic activity, even organs like the bladder and uterus have electrical activation where we would pick up interference from, much less the external noise. So one of the issues with this electrogastrography, and why it's struggled to, I guess, gain clinical traction, is due to the, I guess, lack of spatial resolution and lack of robustness of the signal quality. So more recently, high-resolution electrical mapping has been developed and initially translated from cardiac electrophysiology by a guy named Wim Lamers. Our research group subsequently came and built on that by developing custom printed circuit board or flexible printed circuit board electrode arrays that encompass 256 individual electrode points. So on this, each of those individual gold contacts is an electrode. We can record from up to 256 simultaneously. And one of the important, or two of the important things about this array is that, one, it's flexible, so it can conform to the surface, the curved surface of the stomach and also the small intestine. And two, it's sterilizable. So what that allowed us to do was translate these high-resolution GI mapping techniques to human studies for largely the first time. And that's what we see in the middle here, so apologies if anyone has a weak stomach, especially since we're just after lunch. But this is an image from in surgery, so this is open abdomen human surgery. This patient had a liver cancer, so their stomach was healthy, but it gave us open abdomen access to place our electrodes on the stomach. The electrodes are placed directly onto the stomach, the serosal surface, and then we passively record the electrical activity occurring from all 256 electrodes. You can see here, this is the surgical setup We've got our electrode cables coming out of the wound. The wound has been closed up to maintain the temperature and moisture during our recording periods. We record for about 10 to 15 minutes, and then we remove our electrodes, and the surgery proceeds as normal. So now that we had recorded hundreds of electrode signals or electrode recordings, we needed to extract each of the individual slow wave activation times, or the activation signals, according to these electrical slow wave events. And from 256 channels for long duration, or say 15-minute recordings at 3 cycles per minute, we immediately generate thousands of individual data points, each of which need to be marked so that we can extract the time information and then also use that time stamp or time mark to then calculate things like activation, velocity, and amplitude. So you can see here an example of 3 electrode recordings. And we're able to work with our group and some of our international collaborators to develop automated algorithms to mark these data. So you can see here that red mark indicates the activation time of each of these slow wave signals. So this sharp downstroke is the activation event of the slow wave. So as that electrical wave passes our electrode, we see an initial upstroke, a sharp downstroke, as the wave passes underneath the electrode, and then we see a gradual return to baseline. So we need to mark each of these downstrokes, and we developed, like I said, an automated algorithm to do so. Secondly, we needed to group the sequences of downstrokes into individual wave fronts. So that's across multiple channels. We needed to say which activation times were associated with the neighboring channels, and we again developed an algorithm to do that, which allowed us to then map the actual propagation sequences across this array. So here you can see what's called an activation time map. So what this represents is the propagation of the wave per 2 seconds as individual color bands. So each color represents the area that the wave is propagated per 2 seconds. So that shows us the propagation of an individual slow wave in time, and then we're able to represent successive slow waves in successive maps. And you can see here, again, some of our analytical algorithms that allowed us to deal with the large data sets that we generated. An activation map, we're also able to plot the velocity maps, representing the velocity, or the direction, as an arrow, and the speed as a color gradient across the electrode array. And finally, amplitude maps showing spatial variations in amplitudes or uniformity based as a color array as well. Now we're able to take that mapping, I guess, one step further and animate these sequences to show a more intuitive visualization of slow wave propagation across the array. And you can see here now we're able to monitor slow wave activity across this 2D patch of the stomach across time. So we've now achieved actual spatiotemporal visualization of these events. And this is from a normal patient or a normal human stomach. So again, these patients aren't fully healthy because they are undergoing open abdomen surgery, and we require that to get our electrodes in place. But we were able to team up with surgeons and map patients that had cancers peripheral to the stomach so that their stomach was healthy to establish baseline gastric electrical activation patterns. And you can see in this recording that these slow waves propagate very uniformly. So there's very even spacing, even frequency of the propagation, uniform velocity across the array, and it's always propagating from the top of the stomach to the bottom of the stomach. Now, with all of these software packages and analytical algorithms that we developed, we packaged them into one uniform software package called the Gastrointestinal Electrical Mapping Suite, and that was packaged with a complete graphical user interface, which allows researchers now to an intuitive way to import their data, analyze that raw data, and really makes analysis of large-scale mapping cohorts feasible. Now, another aspect that we've investigated is adding in the anatomical registration of the stomach. So here you can see successive CT scans that allowed us to recreate the three-dimensional anatomy of patient-specific stomach geometries. And we're able to do that across multiple patients and then link in their actual electrical mapping data onto the stomach to add not only patient-specific electrophysiology, but also the patient-specific gastric anatomy. And that's an ongoing area of research. So now to shift gears slightly into the modeling side of things, we've also linked up, or I guess co-developed in parallel, a number of mathematical modeling strategies, which I think you'll hear more from David Nickerson after this presentation and have some actual hands-on tutorials with some of the modeling. But like you've probably heard from some of the other presenters, we utilize a multi-scale modeling approach, meaning that we're able to model from the cellular level. For example, here, a model of ICC and smooth muscle cellular data. And we're able to mathematically represent each of these ionic currents and, I guess, full cell level data and subcell level data into mathematical models of GI electrophysiology. And you can see here an example of a modeled simulation of cell level data compared to the experimental results showing close correlation of our cellular level modeling. Now we're also able to scale up to tissue level modeling and models, for example here, some of the ICC network properties that are observed in tissue. This is mouse tissue, but it mimics human ICC networks. And you can see two different types. So on the left is our experimental tissue. So this is imaged actual tissue networks. And then we've got wild type on the top, which means normal, and bottom is an ICC depletion, where it's been a genetic knockout. So we've depleted the ICC networks, and we're able to then translate and develop models to recreate these tissue structures, which then allowed us to integrate some of our cellular level electrophysiology data to model actual slow wave propagation through these networks and compare and quantify some of the differences or results of the ICC depletion on slow wave propagation. So you can see now we've got an electrophysiology model spanning both the cellular and tissue levels. Now we're also able to scale that up further to the organ level, and this model in particular was informed from our high resolution mapping data that was taken from individual locations on the stomach. We're able to use this organ level model informed by one of those CT scans to recreate correct anatomical geometry. And we're able to integrate our slow wave data into this model to represent or create a summative model of normal gastric electrical activation. And now this is important when I get to the kind of clinical translation of our research, because this normal propagation serves as our baseline for the investigation into abnormal activities. So you'll see here that in the normal stomach, there's a single pacemaker that's located high on the greater curvature that quickly establishes ring wave fronts that propagate down the stomach. They speed up slightly in that distal antrum region and then terminate at the pylorus. In the fundal region, we see no activity, or it's actually permanently depolarized. We see no propagation into that region, which makes sense functionally because that region of the stomach is mainly functionally an accommodation zone or a storage zone to expand and accommodate for contents in the stomach, but not primarily responsible for the actual breakdown or movement of contents. So this, again, is a model that summates really a lot of our data from a normal human mapping cohort and was very valuable in recreating and visualizing that data set. But we're also able to take that model one step further and translate these organ level potentials to the body surface. And I will note that most or all of this modeling work was done by Ping Du, who's a research fellow in our group. So he translated these organ potentials to the body surface to inform or infer the mathematically correlated body surface potentials that would result from these data. Now this is important because if you remember back to that EGG, one of the problems with that is that we had very noisy recordings and we didn't know really what we were expecting to see or what components of that signal were important. So what this model allows us to do is anticipate the expected signal components from individual locations on the body to better inform the analysis of EGG signals, making it much more likely that we'll be able to achieve, I guess, meaningful analyses or meaningful data from those body surface recordings. And that's a project that Peng currently has in the works as well with a couple of students. So that kind of summarizes a lot of our methodology and development of new techniques that we've undergone in terms of electrophysiological mapping and also, also modeling. I'm not entirely sure what just happened. So now I'm going to change gears a little bit and talk about some of the clinical translation that we've achieved with this work and some of the actual real-world applications that we've looked at in terms of patients suffering from gastrointestinal disorders. So the first disorder that we investigated was called, or is called, gastroparesis, which is Latin for stomach paralysis. So in this disorder, patients suffered debilitating symptoms of nausea, vomiting, abdominal pain, bloating, and early satiety. So that means they feel full very early after eating. Now, another key factor of this disorder is that the stomachs fail to empty properly. So the stomachs remain contents for a long time, and actually because of that, the patients end up being malnourished. They're very, very sick. Some of these patients that we saw in clinic were vomiting up to 8 or 10 times a day, and it's completely debilitating to their quality of life. Now, unfortunately, there are very few clinical treatment options for this disorder, and it's not even known what causes that, or the actual underlying pathophysiology remains unknown. So we were interested in investigating if there was an underlying electrical or electrophysiological component of this disorder. So here is just an example patient. So it's a 36-year-old female. She's type 1 diabetic, so there's a close link between gastroparesis and diabetes. She was diagnosed with gastroparesis and actually had to undergo C-section during her pregnancy because the gastroparesis was so severe. So again, it's a very debilitating disorder. Like I said, one of the key aspects is that your gastric emptying is delayed for these patients on top of the severe symptoms. And so here you see the standard clinical test to diagnose gastroparesis. After they've excluded every other possible explanation for the symptomology, they will perform a gastric emptying test where the patient eats a standard meal of 2 radio-labeled eggs. Those show up opaque on the X-ray, and you can see on this slide the time delayed emptying of the stomach. So from here, immediately after the meal to about 2 hours later, we see that most of the meal is now left the stomach and is in the small bowel. So this is your rough stomach outline. We can see that the meal starts to push into the small intestine at about half an hour, and after 2 hours, most of the meal is in the small intestine. So the diagnostic criteria is that after 4 hours, less than 10% of your meal will remain in the stomach in a normal functioning stomach. Now, in this particular patient that I just showed you, this is her scan after 90 minutes. Almost all of the meal was still remaining in her stomach. And I don't recall the data at 4 hours, but it was greater than 10%. So she was diagnosed with gastroparesis. So we teamed up with a clinical hospital in the US that treats a lot of these patients, and they treat them surgically. They implant an electrical pacemaker into their stomach. Now, what that allowed for us to do was piggyback on that surgery and map even just the baseline electrical activity in these patients before they introduced any sort of clinical intervention or surgical intervention. And we did so using that electrical mapping techniques that I explained earlier. So in this cohort, there were 12 patients. All of them were diagnosed with gastroparesis. Medically refractory means that it didn't respond to typical treatments or typical treatment strategies, so it was unresponsive medically. And you can see some of their cohort data here. So importantly, they had median gastric retention of 26% after 4 hours. So again, their stomachs remained full for a long time. And their total symptom score, which is a quantitative metric of their symptom severity, was 16 out of 20. So that's very severe symptoms of nausea, vomiting, bloating, pain, etc. Now, just to remind you, in the normal stomach, this is the general activation pattern that we see. I showed this to you earlier. So we see activation is always oriented down the stomach, very routine velocity, frequency, wavefront spacing. Now, in this cohort of gastroparetic patients, what we saw was a range of electrical abnormalities. So here's an example from one of these patients of an abnormal initiation event that's unstable in the distal stomach. So those first couple of waves that we colored blue appear as though they're normal. They're propagating down the stomach in the correct fashion of similar frequency and velocity that we would expect. But then we see these very abnormal chaotic activities break out in the distal stomach. We see retrograde propagation, colliding wave fronts, multiple wave fronts at once, abnormal initiation in this distal region of the stomach. So very chaotic abnormal activity. Here's another abnormal initiation event from these gastroparetic patients. In this case, it's a stable initiation. So we see activity from this distal region of the stomach. So this is where our electrode recording array was. We're right in that distal stomach. And we see an ectopic pacemaker located on the greater curvature, resulting in retrograde propagation up the stomach. So again, complete opposite direction of propagation as in the normal case. And from that total cohort of patients, we observed that 11 out of 12 of the patients exhibited some form of conduction abnormality. And we broke those down into abnormalities of initiation and conduction and developed this whole classification of gastric electrical dysrhythmias. Now, I don't think any of you mentioned that you had a background in cardiac or cardiology, but this is quite interesting because this mirrors a lot of what was done in cardiac electrophysiology, say 20 years ago, where they've now developed a whole platform of clinically useful mapping and therapeutic techniques, where none currently exist in the gastric field. Now, we're also able, just to bring some of the modeling back into this presentation, to recreate some of these abnormal dysrhythmias in modeling form. So we can see, now we can compare a normal electrical activation pattern on the left, again, in a patient-specific organ geometry, to an abnormal electrical activation of an ectopic pacemaker located in that very distal antrum on the right. So next we're interested in looking at a second clinical disorder. So this disorder is called chronic unexplained nausea and vomiting. And it's interesting, or particularly interesting to us, because it's been shown to have exactly the same characteristics and symptomology as gastroparesis. However, in these patients, their stomach empties normally. So they have normal gastric emptying times, but they exhibit the exact same symptomology of these very severely sick gastroparetic patients. So we were interested in investigating whether these patients also exhibited the abnormal electrophysiology that we had observed in the gastroparetic patients. So in this case, our cohort was 9 patients, similar age range to the gastroparetic cohort, similar symptom score to the gastroparetic cohort, but you'll note that their retention was under 10%. Median retention was 4% in our cohort here. And again, we, or I guess in addition, in this study, we also mapped a further 9 controls on top of the 12 that we had previously mapped, just to basically re-verify that in these control patients, in the healthy stomach, the activity is purely normal. So we did find that in all of our controls, we had purely normal activation patterns, but in the patients suffering from chronic nausea and vomiting, we saw, again, a range of very abnormal electrical propagations. In this case, you can see an abnormal initiation. It's located high on the stomach this time, but more towards the lesser curvature or middle of the stomach, rather than that greater curvature side, which is always the location in the normal stomach. Another example here, we found what's called a reentrant conduction pattern. So again, we can draw close ties with cardiac electrophysiology, where this is widely studied and is one of the leading causes of sudden cardiac death. It's associated with a whole range of cardiac disorders. But this is the first observation in the human gastrointestinal tract. And what we see here is that we get our activity that propagates in basically a looping circuit around this distal portion of the array, reactivating that same circuit over multiple cycles. It results in retrograde propagation up the stomach, which collides with an antegrade wavefront coming down the stomach. And again, very abnormal conduction patterns compared to the normal stomach. So we're able to update our dysrhythmia classification scheme for these patients. And it's important to note, both in chronic nausea and vomiting and also gastroparesis, that a number of the patients exhibited these spatial dysrhythmias with normal frequency. So in this frequency graph, anywhere we see N means normal, and we can see that there were a number of patients across this cohort that exhibited spatial dysrhythmias at normal frequencies. Now that's an important finding, because using some of those lower resolution techniques like they used historically, and continue to use, like body surface EGG, that primarily rely on frequency-based analysis, we would likely miss the dysrhythmic patterns in these patients. Previously, they classified dysrhythmias based almost solely on frequency, whether it was within the normal or abnormal frequency range. And in these cases, all of these patients with normal frequency would have likely gone undiagnosed using those less sophisticated methods. Now, we also added in cellular analysis to both cohorts, the gastroparetic patients and the chronic nausea and vomiting patients. And you can see here an example of the stained ICC networks in a chronic nausea and vomiting patient on the left and a control patient on the right. So in these images, the red are ICC cells and the blue are all of the cellular nuclei. So we actually teamed up with some collaborators at the Mayo Clinic in the US to analyze these images for us, or to obtain and analyze the images for us. And they've got a very sophisticated, or relatively sophisticated, algorithm where they only count the locations of, or co-location of, a cellular ICC body plus nuclei, so that we only count the cellular ICC bodies. And that's important because these ICC networks are in 3 dimensions and have long finger-like extensions, that when you segment them in 2-dimensional space, you can end up counting multiple, I guess, islands of what appear to be ICCs that are actually linked or extending from the same ICC body. So we're careful here to count only the ICC bodies, and quantified those and compared between the disease states and the controls. And what we found was very interesting here, because not only did we find ICC depletion in both of the gastroparetic and chronic nausea and vomiting cohorts in combination with our electrical abnormalities, but we actually saw a graded response of ICC depletion. So the chronic nausea and vomiting patients were depleted, but the gastroparetic patients were even more severely depleted. Now that's interesting, because functionally or symptomatically these two patients are similar. Electrophysiologically these two patients are similar, and they both exhibit ICC depletion. But functionally, this cohort of gastroparetic patients have stomachs that fail to empty properly, whereas the chronic nausea and vomiting patients have stomachs that do empty properly. So it appears as though these two, what are now distinct clinical diagnoses, may actually exist within a spectrum of the same disorder, and are actually two or potentially partitioned patients from the same disorder based on a threshold of ICC depletion that ultimately results in functional abnormalities of the stomach. So that wraps up the baseline work on investigating differences in both gastroparesis and chronic nausea and vomiting. So lastly, what we're interested in studying in the clinic was the effects of gastric stimulation on gastroparesis. So one of the clinical treatment options, and the option that is being surgically implanted in the center that we linked up with to make those baseline recordings that I just showed you, is to implant a high-frequency electrical stimulator into the stomach, which is commercially available and made by Medtronic. So you can see on the bottom here is a meta-analysis study which basically looks at the results from a whole range of academic studies, pools them together, and the take-home message is that the general body of literature suggests that this high-frequency Entera stimulator improves the symptoms in patients with gastroparesis. However, the actual underlying mechanism of action of this device is unknown. So we aimed to investigate the electrophysiological effects of this high-frequency GES, is gastric electrical stimulator or gastric electrical stimulation, including spatiotemporal dysrhythmia response using our high-resolution mapping analyses. So again, we teamed up with that same medical group in the States that were implanting these, and this time we looked at the data post-implant. So previously, what I showed you is all the baseline data before they implanted any stimulator. They then implanted the stimulator, and we mapped the response after the stimulator was turned on. Granted, the limitation was that we were still performing these recordings intraoperatively, so it was the response immediately after stimulation was turned on. We looked at 8 patients, all with gastroparesis. Our median age and total symptom score were similar to our baseline values that I just showed, and our median retention, again, was abnormal. Now you'll note here, and I guess this is the interesting aspect of this high-frequency stimulation protocol, is that the actual stimulation frequency is relatively high or very high when compared to the frequency of our slow-wave events in the stomach. So our slow-wave events in the stomach, you could even tell from some of those animations, operate very slowly, as the name suggests, and they occur at about 3 cycles per minute. Now, this stimulation is occurring at anywhere between 14 hertz and 130 hertz in the 4 different experimental protocols that we tested. So it's not designed to have a one-to-one correlation pacing the slow wave activity. And what we found was that there were no differences in slow wave activity between baseline recordings and any of the 4 stimulation settings that we tested. So those 4 stimulation settings were on the previous slide of increasing frequency, increasing amplitude, and increasing pulse width. So again, there were no changes to propagation pattern. There were also no changes to our slow wave quantitative metrics in terms of slow wave velocity, amplitude, or frequency between our baseline recording and any of the stimulation settings, or between any of the individual stimulation settings themselves. And there was no change to the occurrence of dysrhythmias between when stimulation was off and when stimulation was on. So we see an example here of an ectopic pacemaker dysrhythmia located high or on kind of the mid-region of the stomach in the center line or near the center line or lesser curvature, and that persisted from baseline into and through stimulation. Now, across the cohort, dysrhythmias were observed in half of the patients. They included the standard kind of forms of dysrhythmia that we had previously classified in our baseline data. But importantly, the occurrence was similar between periods when stimulation was off and on. So it appeared that not only did this device not change slow wave activity or quantitative characteristics, but it also did not change the occurrence of dysrhythmias. However, our cohort agreed with a large body of literature that these patients reported improvements in their symptoms post-stimulation implant at 6-month follow-up. So it appears that the stimulator is doing something, but it's not reorganizing or improving the electrical abnormalities that we've observed in these patients, suggesting that it likely functions through a neurally modulated pathway. We also added some current distribution models to explain some of these effects, and also modeled the cellular response from these effects, which is, again, ongoing research. So that kind of wraps up the overview that I was going to give you, or am going to give you, on our platform of GI electrophysiology. So looking more towards the future directions and horizons, we're very interested in now translating towards diagnostics and trying to improve the clinical applicability of some of these methods. For example, by improving the accuracy and robustness of bedside tests like EGG, using some of our modeling data. Translating our electrical mapping approaches to endoscopic delivery so that it can be minimally invasive and applied to a much larger cohort in clinic. And also developing a lot of the device and software infrastructure behind clinical translation of these methods. And we're also interested in looking at pioneering new therapeutic options to modulate these gastric dysrhythmias. So I showed you that high-frequency electrical stimulation appeared to have no effect. However, potentially a lower frequency, higher energy pacing approach closer to that native frequency of slow waves may be able to entrain these abnormal electrical patterns. Or something like ablation, which is controlled tissue burn to induce, localized electrical conduction blocks at sites of dysrhythmia, may be applicable as well. And that's a technique that's widely used in cardiac electrophysiology, but has not yet been trialed in the gastrointestinal tract, although I have now run a cohort of pilot studies with quite promising results using that method. So lastly, I'll just end with an overview of our GI team. So I got the fortune of presenting work that was done by a lot of people over many, many years. Some of those people are shown on this slide, not all of them. But we also benefit, I think, in our group particularly from close collaborations with clinical or clinicians, namely Greg O'Grady, who did a PhD in our group and remains very closely involved in our academic research, but also is an active lower GI surgeon. Prof. John Windsor, who's an upper GI surgeon and also very involved in our academic endeavors, and Prof. Gianrico Ferrugia, who is a clinician at the Mayo Clinic. In addition to a range of other clinical centers that we collaborate with. And obviously, thank you to our funding sources. So that's all I've got for today. If there's any questions or anything, I'd be happy to host questions and have a discussion before I pass you off to David to do some of the tutorials.
I do,
yep.
But I'm not too sure if I can form a good sentence structure.
That's okay.
Yeah.
I wasn't too sure I could form good sentence structure for this presentation either.
Because it's very interesting information that you have presented. Yeah.
Thank you.
So you have a slide on where you have a low count on ICC cells.
Yep.
In the gastroparesis patients.
Yep. I'll try and go back to that.
One. Yep.
This one?
So yeah, and then later on you mentioned the electrostimulation where you use a higher range of frequency and it has no differences with the control.
Yep.
Then do you think there's a correlation between the low amount of ICC cells here versus the stimulation? And then second question would be, see, I cannot restructure.
No, that's good.
But second question would be, will the stimulation increase or have any effect on the ICC count before or after the stimulation?
Yeah, so that's a great question, and it's actually one of the active areas of research that we're currently setting up and starting to run studies on. So, bang on, you identified exactly one of the, I guess, potential explanations for why that electrical stimulation didn't work, although it's not really at the correct frequency to pace these events. But that is a worry in terms of if pacing will be an effective clinical strategy because we've got these depleted networks. So our hypothesis is that there's likely a spectrum within these patients that have ICC depletion that is not severe enough, or that is, I guess, on the less severe end, where we will be able to pace through those networks and restore normal activation. But there's probably a threshold of depletion through which we will not be able to entrain through that network. So if you think of it kind of spatially, if we have enough connectivity to drive current through that whole network down the stomach, then that may be an effective strategy, pacing. But there is likely a threshold of depletion where we will not have enough connectivity to drive current all the way through the stomach. And so it may be an effective strategy for a subset of patients that are on the less severe ICC depletion side, but potentially not the entire cohort of patients. And then in terms of ICC counts and the effect of stimulation, I haven't seen any data on ICC regrowth. I know that ICC deplete normally with age, and so in ages, I believe 70 plus, they have statistically significant ICC depletion. But I haven't seen any data on any sort of regeneration of ICCs yet. So I don't think that the stimulation would foster regeneration, but I haven't seen any data.
Are they plastic? Are they like, how do you say, like, yeah, plastic?
Yeah, it's a good question. Ultimately, I don't know. Yeah, ultimately, I don't know. It's still a lot of that, I guess, what would it be? Like the early age ICC network growth and expansion research is still being done. There's a lot of work being done on that now, looking at how ICC networks grow and expand in early ages. But yeah, I'm not too sure about the plasticity of the networks.
Thanks.
Are there any other questions or discussion points? General observations? Yep.
In the research you saw us, you manually measure the electrical signals, right?
Yep.
Have you ever done some measurement about the mechanical movement of stomach?
Yeah, this is another great question. I mean, you guys are too smart. So that we are purely measuring electrophysiology. So we're not measuring the mechanical motion. It is, I guess this would go all the way back to that first kind of slide that I showed you, that animation of the contractions. So it is known that electrical slow waves are responsible for defining the contractile pattern. And any contraction is associated with a slow wave, but not every slow wave is associated with a corresponding mechanical contraction. So the ICC are omnipresent. Not ICC. The slow waves are omnipresent. They're always. This will be quicker. So we can see here, I'll just replay this animation in the background of the contractile pattern. So every contraction is generated and coordinated by an underlying electrical slow wave, but not every slow wave generates a mechanical contraction. There are a lot of co-regulatory mechanisms that impact the generation of contractile patterns, including stretch, mechanical sensors, hormonal responses, endocrine system, et cetera, et cetera. So there's a whole range that impact the generation of these contractile patterns. But that, again, is a very interesting ongoing area of research in terms of linking electromechanical data and coupling these data. And that's something where, again, I didn't have it in this presentation, but where we've done some substantial modeling work in terms of trying to link the electromechanical data in combined models. Because I think experimentally, those are very, very difficult studies to conduct because you're not only, because you're modeling, or you're recording multiple variables, and particularly in the in vivo setup, which is where we prefer to do most of our recordings because it's closest to the natural human body awake setup. So it's very, very difficult to do those in an in vivo setup. It is possible, but it's very difficult. And that is somewhere that we're looking to expand to, is to link some of the electrical data with corresponding mechanical data.
So you have the electrodes in there with the stomach contractor.
So that is quite an abnormal amount of contraction. We usually don't see those very large-scale contractions. So I think the reason we got it this time, our electrodes aren't in during this recording. So this is during preclinical endoscopy. So they stick a camera down the throat to make sure that there's no barriers and to kind of investigate the stomach. And in this case, they inflated the stomach. And we think that it's actually that stretch of the air inflation that triggered these contractions to manifest. So in our typical in vivo setup, we see very little contractile activity. And we did actually do a study on that, but we were more interested in the opposite. We wanted to inhibit all the contractile activity to show that our electrical signals were the same in the presence or absence of mechanical activity. So even though we see very little typically, we wanted to ensure that what we were recording was irrespective of mechanical contractions. So ideally, we'd like to do the flip side of that and record with, say, large-scale contractile activity and link that with the with the electrical activity as well. And it's something that we're investigating, but it's quite a complex experimental setup. We do quite a bit of work on mechanical activity as well, primarily further down the GI tract in the colon or the large intestine, where we use high-resolution manometry, so that's pressure sensors. So we've got a fiber optic manometry catheter that was developed by some collaborators in Australia. It's about 1 meter long and has 100 sensors on it, so it's 1 centimeter spacing. And we can stick that up into the colon and then map the pressure waves as they propagate through the colon to try and, again, link it with clinical data. I'll stop that recording. I feel like you guys have seen the inside of that stomach enough. So we try and link those colonic recordings with the clinical data to show correlations between abnormal pressure waves and abnormal contractile responses in patients with different symptoms in clinic, and to locate spatially where those occur.
Thanks.
Yeah, no worries. Are there any other questions or anything? All right, I think I'll pass you back off to David.